Today I will tell you how we plan to arm a patient's own immune cells to potentially cure their cancer, starting with triple-negative breast cancer, or TNBC. My name is Rachel Abbott and I am the CEO of Pan-Cancer T. I have spent most of the last decade developing T-cell therapies at a number of biotech companies. Imagine how you would feel if you or a close friend were diagnosed with breast cancer. Sadly, this will be the reality for 1 in 7 women globally. Then imagine you are told that this is TNBC and your treatment options are limited because it lacks the 3 major receptors that successful breast cancer drugs target. Devastatingly, if your cancer is metastatic and has spread to other parts of your body, you might only have 6 months to live. Current treatments are failing patients with metastatic TNBC, but our lead product offers promise for these women. Our bodies are equipped with an army of immune T cells that can recognize and kill virus-infected cells. Pan-Cancer T's technology can redirect that army to recognize cancer cells. Other so-called adoptive cell therapies, where a patient's own T cells are engineered with receptors that enable them to target cancer, have already been shown to have curative potential. Blood cancer patients remain cancer-free over 10 years after receiving just a single dose of cells. Adoptive cell therapies are now an established treatment for blood cancers, with 6 FDA-approved products. However, specific challenges have limited progress of adoptive cell therapies in solid cancer to date. Many companies focus on a small number of targets, which are not expressed in many solid tumours, including TNBC. Furthermore, solid cancer poses a particular problem, as the hostile tumour microenvironment suppresses immune cells, which limits clinical response duration. Our unique platform provides solutions to these challenges. We have a portfolio of untapped cancer-specific targets that cover solid cancers not eligible for current clinical cell therapies. This includes our lead novel target, ROPN1, not found in healthy tissues, but strongly expressed in 90% of patients with TNBC and melanoma, as well as 45% of multiple myeloma patients. We have a validated T-cell receptor, or TCR, discovery platform to discover proprietary cancer-specific receptors, which we can use to genetically engineer T-cells so they target cancer cells. Our lead TCR, PCT1, is IP protected and highly specific for ROPN1. PCT1 T-cells are highly effective in multiple preclinical models. When mice are treated with mock T-cells, their TNBC tumours continue to grow. In contrast, tumours clearly regress with PCT1 T-cells, which significantly outperform a TNBC standard of care ADC drug. I'm incredibly excited about our next-generation T-cell engineering approach that tackles one of the greatest unmet needs in cell therapy. Our lead TCR co-stim platform provides co-stimulation to T-cells in the hostile tumour microenvironment to enhance response durability. In this murine melanoma model, tumours treated with a wild-type TCR fairly rapidly rebound. In contrast, the duration of the response is clearly extended when the TCR co-stim technology is applied. We have now translated this to human TCRs. This in vitro data shows that, unlike wild-type PCT1 cells, our lead clinical candidate, PCT1 co-stim, continues to kill TNBC tumour cells after multiple rounds of re-stimulation. We are now filing for a patent, and we expect our TCR co-stim platform to generate a lot of excitement because its promise can be applied to multiple TCRs and tumour indications. A number of other companies are developing TCR T-cell therapies, and these big deals demonstrate exciting momentum in the field. However, we believe we are the only player with a dual approach, combining both novel solid tumour targets with additional engineering to resist immune suppression and increase durability. Now would be a great time to come on board as we move PCT1 co-stim through CMC and regulatory approval into a phase 1b/2a trial to generate clinical safety and efficacy data. We are seeking €30 million to achieve this, and also to help advance our pipeline, including programs exploiting targets for colorectal and ovarian cancers, and a second next-generation approach. Pan-Cancer T has an excellent team. With a wealth of experience spanning all aspects of taking T-cell therapies to the clinic, we are well positioned to achieve our goals. We also have a supportive supervisory board and scientific advisory board. Thank you very much for listening to our story. I'd be very happy to take any questions. If you're watching the recording, please get in touch.
I'll be happy to give it a go. Hi, Rachel. Thank you very much for the presentation. Great pitch and great approach. There's several challenges kind of associated with general TCR approaches, right? Just to kind of T-cell exhaustion, heterogeneity of the tumor escape. Can you speak a little bit how you are targeting these? Because I'm sure there's a little bit short in time with the 5 minutes to get to some of those.
Yeah, so I think, our lead target is really great for particularly triple-negative breast cancer and melanoma in addressing those target issues. It is very homogeneously expressed, so more than 90% of TNBC patients express this antigen, but also it's very highly expressed and homogeneously expressed across the tumor. And other T-cell studies have really shown that can really help with clinical responses. And then in terms of T-cell exhaustion, that is what our next generation approaches are addressing. And in particular, the TCR co-stim platform really is showing the T-cells that have got that construct are definitely less exhausted. They express less exhaustion markers. They carry on responding much longer than the wild type TCRs. So yeah, we are actively trying to address both those concerns.
Thank you.
Maybe if I may follow up on Christian's questions, maybe I didn't get the answer there, but regarding the heterogeneity, because this is a bit of a challenge, right? You also show that in your primary indication, 90% the target is expressed, but how you make sure that you will be able to then completely target all the different cells in the tumor microenvironment. So if you can explain a bit more.
So the majority of the cells, when we do IHC staining, you can see that the vast majority of the cells in the tumor are positive for the target. We've also looked at metastatic tissues. So the expression doesn't change really between primary lesions and metastatic lesions. So it doesn't seem to get lost. And yeah, so hopefully as far as I'm concerned, it bears all the hallmarks of being an excellent target.
Thank you.
Could you comment on your competitive advantage over, say, like exciting antibody-drug conjugate technologies?
Yeah,
that
so I think
potentially addressed for this target as well, right?
Yep. Yeah, so obviously, like antibody-drug conjugates have shown some really promising results, and actually there is one for triple-negative breast cancer. But I think they are probably target dependent. And so there's been some really excellent results in certain cancers that have got really good targets. I'm not sure that is necessarily going to be true of all cancers. I think one real advantage of cell therapies is that you can, in theory, give a single dose and it could be curative, which is not the case with any of these other drugs where you have to keep on maintaining doses. So yeah.
But do these cancers typically metastasize?
Yes, and our target population will be patients with metastatic TNBC, but we believe the target is still well expressed in those lesions and can really provide benefit to those patients.
Any further follow-up questions to Rachel?
Yeah, I'll do. I have one more, just maybe without going more into the data, because I'd have many more on the data side. But if you could speak a little bit about your approach, kind of experimental approach for the next kind of 24 months, what are your key activities that you have planned?
Yeah, so our preclinical package, which contains all our safety and efficacy data, is nearing completion. That should be done by the end of the first quarter of next year. And then the real key things are the CMC activities. So we're already in the process of transferring our T-cell manufacturing process to an organisation called NextGen in the Netherlands, and they're getting some good results. We also need to generate clinical grade viral batches for transducing the cells. So that's really the kind of key focus of the next 18 months once we've got the funds. And then the idea would be to go for IND CTA approval and go into the patients, which hopefully we could start as early as 18 months from the point of when we start the CMC, or go to regulatory approval 18 months after starting CMC, and then to the patients about 6 months after that. So kind of 2 years from now, or having adequate funds.
Excellent. Understood. Thank you very much, Rachel.
That's okay. And I'd be happy to answer your scientific questions at some other time if you would like.
Excellent. I'll follow up.
Thank you.
We are not shy, just that we have a lot of questions, but it will take very long time to explain them, I guess.
I'm happy to answer them offline.